Hello, everyone. I think we're probably ready to start. So I'm Jason Hupp. I'm with LI-COR Biosciences, and I'll be the one presenting this, I guess, morning for most people, evening for me. Before we get going, I'll say that there's a Q&A feature down at the bottom. If you have questions during the talk, please submit them there. And then when we get to the end, I'll go through the questions that have been submitted in the Q&A and provide answers. If you just want to send me a message or send a message to one of the organizers, then you can send that through the chat feature, but please send questions through the Q&A. So I'm an application scientist at LI-COR. My job is sort of split. I spend about half of my time in support of our sales group, giving technical lectures like this, going around and training people on how to use instrument systems, installing instrument systems. And then I spend the other half of my time working with our R&D group to develop new applications and new technologies for measurements. I work in the environmental group at LI-COR. And if you're not familiar with this, I'll give sort of a general overview of what we do, and that'll provide a nice lead in to everything else that I'll talk about tonight. So we make scientific instruments, and in the environmental group, you could sort of broadly say the type of instruments that we make are designed to plug in somewhere in the carbon cycle. Most of the instruments we make are used for measuring exchanges of energy or mass somewhere in the carbon cycle. And so what I show here is a picture of an eddy covariance system deployed on a grassland in Nebraska. Maybe not as relevant to horticulture folks, but that's what it looks like here. And this system is used to measure the exchange of carbon dioxide and water vapor and methane and sensible heat between the Earth's surface and the atmosphere. We make other sensors that are used in systems like this and elsewhere that measure radiation, light sensors. Probably most relevant for here, we make instruments that measure photosynthetically active radiation. We make a whole series of instruments that are used for characterizing plant canopy architecture or plant biomass, like a leaf area meter. And then we make a whole host of gas analyzers, predominantly for carbon dioxide and water vapor, but for methane and nitrous oxide. And most important for the folks that are probably listening to me now, we make instrumentation that are used to measure leaf level photochemistry, right? When I said carbon cycle, that was probably the first thing that jumped to most people's mind. And I think to talk about those instruments, it's a really good idea to provide sort of a broad picture of photochemistry. We could synonymously say photosynthesis here, and I could use two examples to show how grossly simplified most people think of this. You know, we have great marketing people at LI-COR. They like to put the word photosynthesis on everything as if it's one process. Or, you know, maybe we've probably all in primary school somewhere seen a little diagram that had carbon dioxide and water vapor on one side and oxygen and sugars on the other side, and a little arrow with light written above it in the middle. You know, those ways of thinking about it sort of gloss over a whole bunch of interesting biochemistry that's under different regulatory processes and interacts with each other that surmise to tell us a lot about what's happening inside the plant. So I have some diagrams that I'm going to walk through to give us sort of a broader picture of what I think of as photochemistry. These aren't in great detail. There's certainly some details here that have been glossed over, but I want to use them to build to sort of the two endpoints of what we actually measure, and then to tell the story of the things we can derive in between. So I'll start with just a simple cross-section of a leaf as I show here. And you can see a leaf, you know, it has two epidermal layers on the outside. One or more of those layers has stomatal pores in it. The aperture's here. Inside the leaf, we have a variety of different cells that contain chloroplasts and some airspace inside the leaf. That airspace inside the leaf is saturated with water vapor, right? So there's all these wet surfaces of cells, and that those surfaces are evaporating water into that intercellular airspace. That sets up a diffusive gradient for water vapor between the inside of the leaf and the atmosphere. And if these stomata are open, we start to see water move out of the leaf. Probably everybody's familiar with that process as transpiration. I'm thinking of it and talking about it here in the gas phase. Maybe many people think of it as like stem flow, but same basic stuff. And then opposite to that flow of water vapor out of the leaf, we have a flow of carbon that we call assimilation. And if the leaf is happy and healthy and into the light, that carbon is flowing into the leaf because there's photosynthetic demand to it, right? It's again a diffusive flux. It's driven by a gradient between the carbon dioxide concentration in that intercellular airspace, which is being manipulated by demand, and the atmosphere. And if the leaf is in the dark, then the carbon flux goes the opposite way. We call that respiration. That demand for carbon inside the leaf in the light is driven by the Calvin cycle, right? And that's the part where we're fixing carbon dioxide and using that carbon substrate to build these more complex organic molecules that the plant uses to store energy. Calvin cycle's not free, right? We have to feed it energy to be able to run it in this direction. And the energy that the plant feeds into it is NADPH and ATP. We probably all learned that in college chemistry or college biochemistry. The ATP and NADPH that is driving that carbon fixation inside the leaf is being produced through an electron transport and a proton motive force established across the electron transport chain in the thylakoid membrane. So we're zooming into my little dark green blobs here. ATP comes from hydrogen ions moving through the ATP synthase, and NADPH is generated at photosystem 1 due to electron transport down the electron transport chain. This doesn't happen for free either. We have to put energy into the leaf to make this happen. And the energy that we put into the leaf comes in the form of light. And I've blown up a light harvesting complex here, or an antenna array, and attached it to photosystem 2, but it's important to note that this really exists in both photosystems. But I'll talk about it in the context of photosystem 2. So in these light harvesting complexes, we have a variety of pigments. The one that we all know of is really important and why we see plants as green is chlorophyll. And if chlorophyll absorbs a photon, my little yellow arrow coming in here, you get it excited. And once it's excited, then it can donate an electron to drive what's happening down the electron transport chain. And to get it into the electron transport chain, it has to go through a reaction center. And that may or may not be directly connected to the chlorophyll molecule that absorbed the photon, right? So it gets transferred through this antenna complex before it finally makes its way to a reaction center and then down the chain. That's not the only fate for that energy. So there's a number of different energy dissipation pathways that happen sort of at the pigment and the antenna complex level. And one of them is this transfer to a carotenoid and moving that energy off to the xanthophyll cycle. That's the primary component of what we call NPQ, non-photochemical quenching. And this is an important process in many plants because it's a means that the plant uses to actively regulate the amount of energy that's going into the electron transport chain. And so it can use it to balance the output of ATP and NADPH. It tends to be a sensitive metric of stress. We use it for a number of different things. The other fate for that energy that's critically important for the discussion that we're going to have here is some fraction of it gets dissipated off as heat. And the remainder of it that doesn't go to one of these other processes gets dissipated as photons at a longer wavelength than what was absorbed. We call this chlorophyll fluorescence. So if you were to filter out the light hitting a plant and look at just what's coming back off of it, you'd see these pink photons. They start around, the actual emission at the pigment level starts around 680 nanometers, but what actually escapes from the leaf is somewhere past 700 nanometers in wavelength. Okay, big detailed picture to tell what is a relatively short story here. And that is that photochemistry is complex. There's a lot of stuff going on here. And when we want to talk about an instrument that measures photochemistry, it becomes immediately apparent that we can't just point one thing at a leaf and have it tell us everything that's happening here. Okay. So the instrumentation that we make targets the endpoints of the collection here. And that is specifically, we make instruments that measure gas exchange. And in reality, we could measure the exchange of a number of different gases between the leaf and the atmosphere. But in practice, the ones that we measure are carbon dioxide and water vapor as our proxies for transpiration and carbon assimilation. And we make instrumentation that measures chlorophyll fluorescence. So instrumentation that's counting those photons that are being re-emitted from the leaf. By measuring these endpoints, I'll show you how we can start to figure out the stuff that's happening in between, and then we'll talk about why we'd want to know that stuff in the first place. So I'll start and talk about the gas exchange measurement. We build steady state open flow through gas exchange systems. And conceptually, they're quite simple. In practice, they're very complicated pieces of technology. But to figure out how we get the measurement that we're interested in, it's pretty straightforward. We take a leaf or some portion of a leaf and we enclose it in a box, and we would call that box the chamber or the cuvette. And the leaf sets in that box. And if it's happy and healthy and in the light, its stomata are open and it's releasing water vapor through transpiration into the air inside that box. And it's taking carbon dioxide up through assimilation out of the air in that box. And the steady state open flow through part of this is that that box is open on both sides, and we have a constant stream of air entering it and exiting it. And that air enters at some molar concentration for carbon dioxide and water vapor and some molar flow rate. And then it exits at some different carbon dioxide and water vapor concentration. And because the concentration is different, it exits at a different molar flow rate, right? We're counting molecules as they move through the box. And the mass balance for a system like this looks pretty simple. You take what goes in minus what goes out, and you divide by the surface area inside the chamber of the leaf exposed inside the chamber, and you can compute the assimilation rate if you're looking at carbon dioxide, or the transpiration rate if you're looking at water. And I just show two forms of the way we do this here. One's the fundamental mass balance, and then one's the way we actually do it. It's maybe not really relevant for here, but the reason that we use it this way, the equations in gray, is because there's no flow sensor that's actually sensitive enough to measure the difference in molar flow rates between what goes in and out of these chambers. Okay, well, this gets us our assimilation measurement. It gets us a transpiration measurement. And we can take these two basic measurements and we can use it to tease apart a whole host of different gas exchange parameters from the leaf. And these are the sorts of things that we report directly from an instrument. And like I just said, this is the way we get transpiration. This is the way that we get assimilation. And the assimilation has a context to it, right? So we can measure the net assimilation, that is the instantaneous measurement we get from the instrument. If we put the leaf in the dark, then we can make an estimate of dark respiration. And if we look at the difference between the net and the respiration in the light, that gives us the gross assimilation, just what's going in. From these fluxes, the transpiration and the assimilation rate, we can apply a fixed first law of diffusion analogy to these. And from that diffusion analogy, we can start to partition the diffusive pathway between the atmosphere and the inside of the leaf to give us information about the limitations to those gas movements. So instead of just quantifying how many molecules of water are coming out and how many molecules of carbon are going in, we can look at things that are related to the stomatal aperture or the endpoints of the gradient to find out more deeper physiological information about the leaf. And the things that we derive, if we're looking at water vapor, would be stomatal conductance to water vapor, or in an instrument that measures CO2, it would be stomatal conductance to carbon dioxide. And if we're measuring carbon dioxide, then we can estimate the intercellular CO2 concentration as well. That's the concentration of carbon dioxide in the intercellular airspace inside the leaf. We can take these parameters even further and derive more things from them, like intrinsic water use efficiency, and we can make estimates of mesophyll conductance, but that's sort of beyond the scope of what I want to talk about here. The other measurement that we make is a fluorescence measurement. And I have 3 slides to summarize fluorescence here, which probably doesn't do it justice because it's biochemically a quite complicated thing to measure, but the implementation and the derivation of the parameters is fairly straightforward. The technique that we use is called pulse amplitude modulated fluorometry. That's the primary methodology we use to make fluorescence measurements. And the fluorescence measurement itself is sort of predicated on this idea, the law, that the amount of energy that goes into the system and is captured by the leaf has to equal the total amount of energy that gets dissipated through all of the dissipation pathways, right? The leaf is not it's not a sink for energy and it's not a source for energy, right? So whatever we get back out of it through either photochemistry, PC here, heat dissipation, fluorescence, non-photochemical quenching, that's the total the amount that went in. And so you can start with that basic premise. And if you think about these things in the context of how quickly each one of these pathways can dissipate the energy, you can define a rate constant for them. And so we could have many pages of derivations here, but I've simplified it to a basic equation that says that the yield of fluorescence, this phi f parameter, which describes sort of, you can think about it as the magnitude of my red arrow here, is the balance of energy dissipation via all of these different energy dissipation pathways. And to some degree, whoever gets the energy is whoever the fastest at that moment in time. And then the other thing that we say here is that the rate constants for non-photochemical quenching and photochemistry, these are apparent rate constants. So we apply a multiplier to them that describes their state. And so those multipliers are Z and A here. And I just picked those variables because it's the xanthophyll cycle that primarily makes up NPQ. And for photochemical quenching or photochemistry, it's controlled by the state of QA. So we start here, and then the technique of PAM fluorometry is just predicated on using different types of light to drive the leaf into different physiological states where one or more of these rate constants is constrained because of the state of their multiplier. And so you can see my top figure, I just have some gray lines that represent light intensity. This first section of the figure, where the gray line is almost on the x-axis, that represents what we'd call a dark adapted measurement. And this is the first part of the modulated part of PAM fluorometry. So in this case, we're applying just a little bit of light to the leaf to get some fluorescence yield, but it's not enough for the leaf to really do anything with it. It's, you know, it's generally less than 1 ppfd, so less than 0.002 of full sun intensity. And then the pulse part of PAM fluorometry, we provide a pulse of really high intensity light, and that drives the leaf into a different state. It causes this rise in the fluorescence yield that you can see in the lower figure. And then we can do the same measurement in the light. The difference here is that in addition to our sort of our background modulated light that we're using to induce some fluorescence, there's actual light that's driving photosynthesis in the background, and that's why the gray line is elevated, and so on and so forth. Each one of these points on this red figure down here represents a different state of this equation up here, a different physiological state of the leaf. So in the dark, when we're just giving that modulated light, you know, NPQ is not induced, so Z equals 0. 0 times K NPQ is 0, right? NPQ is only induced in the light, so in the dark, it's turned off. And then in the dark, in a fully relaxed state, photochemistry is at its potential maximum capacity, right? So A equals 1, there's no reduction in its rate constant. We provide the saturating pulse in the dark, we drive both A and Z to 0. And so in a really simple sense, if you think about the fluorescence measured during that modulated light in the dark and during that pulse, the difference between those, that yield is primarily driven by the state of photochemistry, and it's related to the leaf's capacity to do photochemistry. So this is how we take these fluorescence measurements at different points, at different light treatments, and we start to derive things from them. And to get from this step to the next step is kind of complicated. So we'll just jump ahead and I'll give you some idea of what we can derive from these measurements. So I'm back to my diagram here, and we'll point out some things. So the most widely measured fluorescence parameter, I think I see cited anywhere, is this parameter called FV over FM. It's a quantum efficiency. It describes the efficiency of photosystem 2 at capturing and converting light to doing photochemical work. And the reason that it's so widely measured is because evolutionarily it's highly constrained. If you go measure healthy plant material pretty much anywhere on Earth, this value falls out at about 0.82. And as stresses build in the leaf that degrade the photosynthetic machinery, FV over FM declines. FV over FM would be the parameter that we would derive from those two measurements I described in the dark previously. We make similar quantum efficiency measurements in the light. That's called phi-PS2. It's the operating efficiency or the realized efficiency. It's how good the plant is at that moment at capturing and converting light to do photochemical work. And then we have this parameter FV prime over FM prime, which is measured a little bit differently than what I showed in the previous slide, but it's something that we still do. And that falls somewhere between the maximum potential efficiency, FV over FM, and the operating efficiency. It's sort of how good the leaf could be if we could get rid of some instantaneous stresses. And then we measure things like MPQ, so we can estimate non-photochemical quenching. Like I said earlier, this is a process that's used to regulate the outputs of the electron transport chain. It's a process that's upregulated in response to stress or in response to excess light. We can make estimates of the state of the reaction centers. So this parameter QL tells us how many the fraction of open reaction centers, or 1 minus QL, which tells us the fraction of closed reaction centers. And then from the phi-PS2 measurement, if we know something about light absorption by the leaf, we can estimate how many electrons are going down the electron transport chain. And this is a neat parameter because there's some basic stoichiometry that sets a fundamental limit for the number of electrons it takes to fix a carbon. And so by coupling these fluorescence measurements with the gas exchange measurements, we can really paint a really deep picture of what's happening physiologically inside the leaf. So then the question comes, why do we care about measuring these things? Well, nothing's free, right? Plants use these processes to capture light and convert it into the food and the products that support their growth and productivity. Without these processes, the plant doesn't grow, it doesn't exist, it just dies. And so measurements of them can be predictive of broader scale or longer term performance. There's quite a bit of literature that shows that certain physiological parameters can be related to yield, right? Because this is the carbon capture that supports it, and the energy capture that supports that carbon capture. There's probably 40 years of, maybe 50 years of literature now that describe all of these various relationships, and I'm not going to go into them. But I will say that the reason that we want to measure them, that we don't want to just read about them in the literature, is because their capacities, their instantaneous behavior, these aren't static properties of the plant. These things change phenologically, they change ontogenetically, they change in response to the environment. And that's both thinking about sort of like the broader scale environment around the plant, but also sort of smaller scale environments that happen within a plant canopy, right? And probably for the purposes of most people listening here, we care about these things because they are dynamic. They respond to the environment. They respond to the state of the plant. So they can be predictive of plant health. These can be used for detection of stress, the detection of recovery from a stress event, for evaluation of how much recovery happened, and for evaluation and elucidation of the physiological mechanisms that underlie the stress response of the plant. Right? So they're really valuable parameters. So that's the pitch to get here. Now let's talk about what we make at LI-COR to do this. So we make two combined gas exchange and fluorescence instruments. We make the LI-600 porometer fluorometer, which is the young lady in the picture is using. And then we make the LI-6800 portable photosynthesis system, which the young gentleman is using in the picture. And I'll talk a little bit about each one of these and the kind of things that they're really optimized for and can be used for, and then make a pitch for how they can be used together. So the 600 porometer fluorometer is, as its name says, it's a porometer fluorometer. So this is an instrument that measures water vapor. And so we can derive apparent transpiration from it. We can derive stomatal conductance to water vapor from it. It includes a fluorometer. It's a fairly basic PAM fluorometer because it's an open fluorometer. And so we can derive FV over FM and phi PS2 and ETR from its measurements. You can jump through a series of hoops with it and get at NPQ, but it's a little bit more difficult. This is an ambient instrument. So the fluorometer, the leaf is open. Give me a second to move this thing. So the leaf sets in this little clip right here. And so the leaf aperture that measured is measured as this little circle. It's about 7 millimeters in diameter. The lower part of the leaf, this part sets below the lower part of the leaf, and that's where we make the conductance measurement. So we're getting the conductance on the bottom side of the leaf. It's a single-sided measurement. And then the fluorometer sets viewing the leaf, and it uses ambient light to illuminate the leaf. And then we have some LEDs here that we use to provide the modulating and flashes, and the fluorescence detector sets in the middle. The instrument includes an ambient PAR sensor. This measures photosynthetically active radiation. Power for the instruments in the handle. It has onboard data storage. We access charging for the integrated battery and data through a USB port on the back. And then, as you can see here, there's a user interface on it that we use to interact with it. This point about it being an open fluorometer, an instrument that makes measurement under ambient conditions, is a really important feature of this. So the 600 is an instrument that's really optimized for what we would call a survey application. And a survey application or a survey measurement, these are snapshot measurements collected under the current environmental conditions the leaf is seeing. And they tell you what the leaf is doing at that moment in time, right? You know, I said earlier that these photosynthetic parameters, they're not static. You know, there's this whole host of developmental things that changes them, but there's instantaneous behaviors that happen that cause them to change over the course of a day. And so you can see on the right-hand side here, we have two figures. This is phi PS2 and stomatal conductance to water vapor measured with the 600. That's what the solid symbols are. And these measurements were made across a canopy of a North American native hibiscus. And you can see plotted with this in gray is the instantaneous light intensity. This isn't from the 600. This is measured with a sensor that's recording continuously outside of this canopy. And this isn't a perfect day to illustrate this kind of behavior, but it works. There's a lot of clouds that pass over, and that's what the dips in light intensity are. But you can see that there's this trend in both phi PS2 and stomatal conductance to water vapor that are driven by light, right? Because, well, what's phi PS2? Well, that's our metric that's related to electron transport, which is related to carbon uptake. That electron transport is driven by light intensity on the leaf. That carbon uptake requires there to be an open diffusive pathway between the leaf and the atmosphere, and that diffusive pathway is controlled by the stomatal app. So if the stomata open to let carbon in, they have to open to let water vapor out. And this would be a little bit clearer on a better day in the conductance and the depression. And so a lot of times we'd think of this conductance, you know, because it's related to carbon capture, that it would follow the light intensity throughout the day pretty closely. And if light was bell-shaped, it should be bell-shaped too. But lots of plants show this dip, and that represents sort of short-term accumulation of stresses and biochemical feedbacks inside the leaf that cause things to down-regulate photochemistry, even though there's plenty of light to do it. The other thing that makes the 600 a really optimized instrument for survey measurements is that it's fast. It's certainly the fastest instrument we make for any of these kind of measurements. I show some data on the right here. These are collected on conductance standards. So these aren't real leaves. That's why they give exactly the same answer on repeated measurements, because there's no physiology to change. The bottom panel is for a 0 standard, which is a Teflon coated piece of stainless steel. And then the top is for an artificial leaf that's composed of a water diffusive membrane and a reservoir and is temperature controlled. And the way I collected this data, those dips are where I opened the chamber. And I open the chamber for 3 seconds or so, enough time for it to wash out, and I close it. And then I'm recording data continuously through the PC application for the 600. And you get these sort of recovery times as it comes back to steady state. And it doesn't really matter how long the chamber's open here. That recovery time is driven by the kinetics of what's actually happening inside the instrument. And you can see it's pretty quick. I mean, we're less than 10 seconds to full stability on these things. And then those red and black data points are marking where we have some default stability threshold on the instrument. And those would be the points where if you were using one of those default thresholds, that the 600 would have logged data for you. And it's worth noting that here on that top panel, they're within 1% of the full steady state value. So they're pretty good criteria. The point of this is to say that this thing's fast, and being fast does a couple of things for you in a survey application, where the data might be highly variable because of a highly invariable environmental conditions, like on my previous slide. Fast data collection allows for really high data density. And so these environmental variability that feeds back into the physiological parameters becomes a whole lot less problematic, because you can make so many more measurements of the physiological parameters that it averages out. Because it's quick, you can use it in sort of large scale screening programs. I mean, a measurement that takes 5 or 6 seconds, it's not difficult to screen several hundred plants in a day. The other reason that it being quick is really important is that because we make the measurement so quickly, we can make it before the plant has a chance to respond to any changes we might induce in its environment, right? So part of the 600 measurement is increasing the turbulence of the leaf surface. And by making the measurement quickly, we don't give the plant a chance to respond to that increased turbulence. Most plants don't respond to turbulence, but a few do. The other thing that we do in the 600 that makes it really nice for these sort of large scale screening programs and high data density measurement schemes is we include some nice features in the instrument for data curation. We support some user configurable prompts, and you can pre-configure those and select them from a list, and that kind of works well. But the thing that really works great is it has an integrated barcode reader. Ooh, excuse me. On the bottom of the instrument here, there's a little barcode reader. You can see the pink light from it here. And so as you go out and make the measurements with the 600, you can hit the button to record the stomatal conductance and the fluorescence parameters. And then you tip the instrument up and you can have all your plants or pots barcoded, and then just scan those barcodes and it will automatically write it into the data files. And so here's a screen capture from one of the data files, and you can see that I was using barcodes for this and measuring sunflower, and that was the species that the barcode translated to. The 6800 is a little bit different. It is a full portable photosynthesis system. So unlike a 600 that just measures a subset of the fluorescence parameters and then the water vapor related gas exchange parameters, the 6800 measures both water vapor and carbon dioxide and has a full featured fluorometer. So it can provide all the fluorescence parameters that I've described. It can actually do additional fluorescence parameters that I haven't described here. It's set up to do induction kinetics, as well as derive a few other things that I haven't talked about. But like the 600, there's a leaf clip or a chamber. In this case, we enclose both sides of the leaf, so we're measuring an integrated value from the top and the bottom. And there's a light source above that clip, and the light source that I show here has the fluorometer integrated into it. We still have an ambient PAR sensor for measuring photosynthetically active radiation. The instrument itself, instead of being one handheld device, is two sort of easily field portable devices. There's what we call a sensor head, and that contains this temperature control module and most of the gas exchange components that I have drawn out here. And then there's a console that sits back here, and that contains the power supply and the data storage. And you can't see it here, but there's a touchscreen user interface on the top of the instrument. And then it includes the air conditioning components. And that's what really sets the 6800 apart from the 600. So you can use a 6800 to do survey style measurements. But the 6800 doesn't make measurements under the ambient conditions. It's a fully user-controlled environment inside the chamber. So if you want to do a survey style measurement, then you get to set the chamber environment to the ambient CO2, the ambient water vapor, the ambient temperature, the ambient light intensity, right? Because we control all of those things. You have full control of the CO2 concentration at the leaf, the temperature of the leaf, the vapor pressure deficit or the humidity between the leaf and the atmosphere. And you have control of the light intensity from something from 0 to well above full sun intensity. And the light source in the fluorometer is a red-blue light source, so you have some control of spectral quality as well. This means that you could also just set the 6800 chamber to some arbitrary set of conditions. And what I mean here is the kind of conditions that we would use to measure physiological response to the environment, a response measurement. So to do these kind of measurements, we set the 6800 chamber, all the environmental parameters to fixed conditions, some fixed set of conditions, and then we pick one of those environmental parameters and we iterate over it. And I show two examples of that iteration here. The top figure plotted in blue, we'd call a light response or an AQ curve. And the bottom figure plotted in green, we'd call a CO2 response or an ACI curve. And so in the top figure, we have carbon assimilation plotted over light intensity. And you see this very typical shape of the response. In some plants, this response doesn't reach an reaches an asymptote and then starts to decline. But this is typical of a high light adapted plant. I probably collected this on field bean. And we would fit these curves to find out a whole bunch of physiological parameters that we can't tell from just an instantaneous measurement of the physiological parameters alone. So if we look at the asymptotic assimilation rate here, we would call that A max, or sometimes you see it written A sat. And that's the light saturated assimilation rate. That's how much carbon the leaf can take up per unit time if light is not limiting the photochemistry. From the y-intercept, we get the dark respiration rate. From the x-intercept, we get the light compensation point. That's the minimum light intensity the leaf has to have before it can start providing some net accumulation of carbon dioxide. We can figure out what a saturating light intensity is, what that maximum intensity is that the leaf can deal with before there's no gain in carbon assimilation. If we're working with a leaf that saturates at a lower intensity, like I said, these curves will start to tail back down. You can find out at what light intensity you start to see photoinhibition of photosynthesis. There's too much light and the leaf has to start downregulating things. The lower panel, that's carbon assimilation plotted against the CO2 concentration inside the intercellular airspace, the CI. And we fit these curves with a couple of different models to derive what are fundamentally enzymatic efficiencies. So this maximum rate of carboxylation by Rubisco VC this parameter called J-max, which is related to the maximum electron transport rate, which sets up a limitation at how quickly the leaf can regenerate RUBP. If we take it out far enough, we can make an estimate of triose phosphate utilization. We can fit a demand function to this that gives us a parameter called stomatal limitation. And stomatal limitation gives us information about how much more photochemistry the leaf could do if it didn't have the stomata separating the inside of the leaf from the atmosphere. And so we use these response measurements to explore physiological capacities and understand physiological limitations. So this all sounds exciting, 2 cool instruments. And they're very useful in their own right. So there are clearly use cases where somebody might only be interested in response data. There are clear use cases where somebody might only be interested in survey type data. But these 2 instruments actually make a really powerful tool if you use them together. The 6800 measurements are considerably more complicated than the 600 measurements. And for a number of the measurements we make in the 6800, the actual mathematical models that we use to derive the parameters require a number of inputs that are not measured things, right? So the best example that comes to mind of this is, so we make a 2-sided measurement for the gas exchange measurement in the 6800. And so to be able to derive parameters like CI, or stomatal conductance to CO2, or stomatal conductance to water vapor, we need to know something about how stomata are distributed on the upper and lower surfaces of the leaf. So we include a parameter in the model that's actually used to compute those things called stomatal ratio that describes that. And there's lots of ways to figure out what stomatal ratio is. You can do epidermal peels and count stomata on one side of the leaf versus the other side of the leaf, but it's tedious. You could look in the literature, but that may or may not have anything to do with the plant that you're actually measuring. But you can take a 600 and you can make measurements on the top and the bottom of the leaf separately. And you can make a reasonable estimate if you're measuring a healthy leaf in a fully engaged state at what the stomatal ratio is. And so you can use the 600 in a number of ways to parameterize the inputs for the 6800 calculations. Maybe the bigger use of the 2 of them is that these data, if you think about it in the context of, say, a large-scale screening program that you might use to say, maybe it's a production situation and you have hundreds of different genetic lines and you're looking for some particular behavior, maybe a particular stress resistance, or maybe you're looking at the ones that are most sensitive for a particular stressor. We might even extend this a little further and talk about it in the context of managing production. But the idea is that these sort of screening programs can be targeted in sort of 2 different ways. I think about it as like the discovery management divide. And so in a discovery context, that's like, I have hundreds of different genetic lines. I'm going to use these rapid measurements with the 600 to be able to rapidly screen these different lines for some particular physiological parameter or group of physiological parameters that are related to something that I'm looking for. Maybe I'm looking for drought resistance. And so I'm just going to screen for stomatal conductance. And I'm going to go measure these things early morning and mid-afternoon. And it's a 5-second measurement on both. And so I can collect a whole bunch of data. But then I want to understand what the consequences of that drought stress response are when I finally find, the individuals, the lines in this broader group that show, maybe they show the strongest stress response and maybe the ones that show the least stress response, just looking at conductance. And then we might apply the 6800 to do these sort of detailed response measurements, to look at like stomatal limitation, to look at the consequences to light response or to CO2 capture, or to do something like a vapor pressure deficit response, and to try and tease out sort of the physiological consequences of what happens deeper in the leaf when that stress engages. Now, in a lot of applications where I worked, for example, before I came to LI-COR, was in a crop modeling lab, we would take that deeper physiological understanding and we would use that to parameterize a crop model. So we use those response measurements to build a predictive model that describes, given a set of environmental parameters, how the plant will respond. And then we might use that model to pick out thresholds for a particular physiological parameter that we're going to use to guide our production. We know that if, I don't know, FV over FM drops below this particular value this many days after we stop irrigating, that if we let it go further, it's going to have this significant consequence on yield. But if we irrigate at that point, then maybe our yield gets restored to some significant fraction of the original potential. These are just sort of off the cuff examples. And the reason this works is because these survey measurements and these response measurements, we're measuring the same thing in different states. They're related to each other. And I think sometimes we have a hard time grasping that because there can be so much biological influence on the values that we see for a survey measurement. And so I have one data set here that I just want to use to illustrate that before we move on. And so these are light response measurements. In this case, I'm plotting electron transport rate versus light intensity. And this was done on okra. And the solid symbols were measured on a single leaf using a 6800 in a fully controlled environmental chamber, right? So I took one leaf, I clamped it, I made a response curve measurement. The open circles I measured with the 600 on the same canopy, but I measured leaves that were already setting in different light environments. And then I just plotted one data set on top of the other. And you can see that this response measurement that I make on the single leaf level, it's related to how the whole canopy is behaving. And the measurement that I get for a given leaf setting in different light environments is related to what I could predict how that leaf would behave from that detailed light response measurement. So the two are intertwined, and that's why this discovery management type scheme of interleafing that data works so well. So this is pretty much my full pitch. I have two slides here. One is that if you want to learn more about the particular instruments, obviously you can find these on our webpage. So there's the links for the 6800 page and the 600 page. If you want to understand more about the physiological parameters I talked about, we have a really nice document that was put together for the 6800 called Defining the Value of Photochemistry Parameters. And this document walks through sort of each one of these parameters. It gives the notation we use for it, the units that it's measured in. It gives a short description of what it is and sort of what it means physiologically. And then paired with each one of these is a couple of key references from the literature that either describe the physiological basis of the parameter or techniques for measuring that parameter. And then my concluding slide, I'll say thank you. I thought this was funny. I hope this has been illuminating. So I put one of our spectroradiometers on here used to measure light. And if you have questions after this, you can please contact our ENV sales at the email address below. And at this point, I can start going through the questions that were submitted during the talk. So I will stop sharing my screen so that I can see the Q&A. So there's a couple of questions here. So one is, if the trees we want to measure are so high, can we cut the leaves down to do the measurement? And how long after being cut down, the leaves are still good for measurement? And this is from somebody that's already using a 6800. Yeah, I think there's fairly well-established protocols for this. So a lot of people that do tree physiology do, well, I learned this from a tree physiology. He used to call it shotgun physiology because when he was in graduate school in like the 60s, they would take a shotgun out to the field and they'd shoot the branches down that they want to measure. I don't think anybody in the world actually does that anymore, but it's kind of humorous. But the point is that, you know, for really tall trees, a lot of times we do have to cut the branches. And it's really interesting. Some plants have near instantaneous response, near instantaneous wound response to conductance. And some plants have a really slow wound response. And so if you're going to do this, it's very typical for people to cut branches, but the data set that you have to collect before you're going to collect your real data for publication is you're going to have to collect some measurements in the field with the branch attached. Then you're going to have to cut it. Typically, you'd put it in water and recut it again to reestablish that transpirational stream. And then you'd make a time series of measurements after that to look at how quickly any physiological response from that cutting sets in. And typically you wouldn't publish this, but it is something that a lot of reviewers will ask for when you go to submit something. And then there's the question, is there a way to calculate transpiration rate from stomatal conductance to water vapor measured by the 600? It's actually the other way around. The first thing that we do is we calculate the transpiration rate. And from the transpiration rate, we derive the stomatal conductance to water vapor. So we do report a transpiration rate in the data files with the conductances. But I always call this apparent transpiration rate because it's the transpiration that's happening inside the chamber. And so that transpiration rate will be quite likely different than what the leaf was doing before you put it in the chamber. And this is just a consequence of Fick's first law of diffusion. And it's because the chamber environment and the leaf temperature are going to be slightly different in the chamber. So chamber humidity is going to be slightly different than ambient. Boundary layer conditions are different in the chamber. Leaf temperature is a little bit different. So the things that are making the endpoints of the diffusion gradient that drive transpiration are different. And so for people that are interested in transpiration rate, the way that I like to think about it is that conductance is the transpiration rate standardized by all of those sort of abiotic things that make up transpiration. So you could say E divided by evaporative demand is GSW basically. And if you think about it that way, then conductance is great for making comparisons between measurements. And then if you know something about evaporative demand in the field, then you can take that conductance measurement and estimate transpiration from it going the other way. And then there's a question, in dark adapted leaves, can I affect the measurement of GSW? Maybe. Generally in the dark, the stomata close down. In fact, stomatal closing as you cut light happens pretty quickly in most plants. And we generally think in a fully adapted leaf that the stomata are as closed as they possibly can be. And stomata are typically really slow to open. So generally closure happens on tens of seconds to maybe minutes of time scale. And stomatal opening generally happens on tens of minutes of time scale. And so there's a major difference in magnitude of response speed. So if you start with a fully dark adapted leaf and you're trying to get like minimal conductance in the dark, then so long as you don't put the leaf in the light, then, or if you put it in the light for not very long, you'll generally get a reasonable measurement. Then there's the question, can I measure FV over FM in dark adapted leaves with the LI-600? And the answer is absolutely yes. It's an ambient light measurement though. So if you want to do dark adapted measurements with the 600, we either do them pre-dawn or we do them, if you work in like a growth cabinet, you turn the lights off in the growth cabinet for an appropriate period of time to dark acclimate and then make the measurement. And then the next question is, in a vegetable cropping system, surveying using the 600 over time in the system, how important is it to measure at the same leaf height and angle across the survey? Yeah, so I think for survey measurements, it's really important. Well, no, for all measurements, it's really important to try to constrain the potential variability in the measurement as much as possible. And so when I go design a sampling scheme in the field for a variety of leaves, I will control for, if I'm doing it under ambient conditions, sunlight, I'll control for leaf angle. I will control for nodal position of that leaf. I'll control for the position of that leaf in the plant canopy. And then depending on what kind of like plant material I'm working on, I'll control for the spot on the leaf or the spots on the leaf that I'm going to measure. If you think about something like corn or rice, you know, these grasses, the leaf arises from a basal meristem, and so there's an age gradient across the leaf. And in corn particularly, where the leaves are quite long and they last a long time, that age gradient has real physiological consequences. And so I just try to constrain for all of that. And I think that is our last question. So at this point, I think our hosts are going to put up a screen capture with some notes about the next lecture that's going to happen this afternoon. And I will say thank you for listening, and wish you guys all a good day. Thank you.